Good morning students, welcome to the class. So today we are moving on to the next new chapter, that is blood circulation. In this we are going to discuss about the circulatory system. To begin with, we will be learning something regarding body fluids. Okay, so what are the different types of fluids that are present in our body, that is body fluids. So you know blood is the main connective fluid tissue which is present in our body, isn't it? Now the first we are going to discuss about the fluid blood. So when you are seeing this blood, the blood is divided into two parts. One is the cellular parts, which we call it as the formed elements, or we call it as corpuscles. What do we call them? Corpuscles or formed elements. And the other one is your plasma. So the fluid part of the blood is called as the plasma, and the cellular part of the blood is called as the corpuscles or the formed elements. Now these corpuscles, when you are seeing, what are the three types of corpuscles which are present? One is erythrocytes, second one is leukocytes, and third one is thrombocytes. Erythrocytes, you know, it is RBC, leukocytes is WBC, and thrombocytes is what? It is platelets. Okay, so erythrocytes is helping in main function is to carry oxygen and carbon dioxide gaseous transport. Leukocytes are the WBCs which are helping in preventing us from many diseases, as they are acting as soldiers, and thrombocytes are playing a major role in clotting of blood. So when we are getting hurt, if the blood flows, the blood should clot after some time, so that the blood is not lost from our body. So for that, what has to be formed? Clot has to be formed. So formation of that clot is carried out by whom? The thrombocytes. So these are the three types of corpuscles or the formed elements which is present in the blood. Now the next part is your plasma. This plasma, when you are seeing, I have told you, it is a fluid connective tissue. So the major composition of the plasma is what? It is water. So 90 to 92% it is made up of water. The rest you can find some other substances also, like you can find much amount of proteins, much amount of glucose will be dissolved in plasma. Then proteins like albumin, globulin, fibrinogen, many mineral ions will be dissolved in plasma, enzymes are dissolved in plasma, hormones are dissolved in plasma. So like that you can find this plasma is a fluid which is helping in transporting substances from one part of the body to that of the other, and this plasma fluid contain innumerable number of things. Okay, so whether it is carbohydrates, proteins, fats of simpler forms, or not fats, it's corrected by lymph, okay, not by blood. So this is regarding your plasma, that is the fluid part of the blood, containing major composition water, some amount of protein, some amount of dissolved water, minerals, okay, hormones, enzymes are present in the blood. Now when you are looking into the different types of cells, that is erythrocytes, leukocytes, and thrombocytes, there are different types. Like you can find, this is the structure of erythrocytes, this is a WBC, basophil, neutrophil is again one WBC, monocyte is a WBC, eosinophil is a WBC, lymphocyte is a WBC, and this is platelets. So these are the different structures of different types of corpuscles, that is the cellular component which are present in the blood. Okay, so you have a different types of WBCs, then you have platelets, and then you have what? The RBC. Okay, so the shape has been shown, you can just go through the shape how it is, right? Now moving on to the formed elements, the first formed element what we are going to discuss is erythrocytes, that is the RBC. Okay, this is most abundant. If you are taking per cubic mm of blood, means millimeter cube of blood, if you are taking in that cubic mm of blood, you can find what? How much amount of RBC is present? 5.5 millions of RBCs are present. Okay, so in adult man you can find in 1 cubic mm of blood this much amount of RBC is present. That's why the blood is having what? Red color, as that is the major composition. Now where is this RBC formed? It is formed in the red bone marrow. You know what is a bone marrow, isn't it? So inside the bone there is a cavity, and inside that cavity the fluid is helping in the secretion of this RBC. That fluid which is present inside the bone cavity, we call it as what? The bone marrow. So from there only you are getting what? The RBCs are getting formed. Okay. And now another thing is that when they are formed, they have nucleus. Immediately after they are formed, after some time, the mature RBCs, they do not have nucleus. Why the mature RBC do not have nucleus? Listen students, it's very important. Why mature RBC do not have nucleus? See, if a nucleus is present, then the shape of the RBC should be somewhat round, somewhat spherical in nature, because the nucleus allows the cells to take up its stage somewhat oval or spherical in nature. Okay. Now, when the cell is becoming spherical, so all the RBCs are becoming spherical, means what will happen? You know, blood vessels like arteries, veins, it is okay, the RBC can move. But when it comes to capillaries, the capillaries are very thin blood vessels, okay, so this much thin they are. So, in case if the RBC is they are spherical or oval in shape, then what will happen is that they will get they will get they will adhere to which one? The capillaries, and they will create a block. Okay, they cannot move, the blood fluid cannot move here as they are creating a block. In order to prevent that, the mature RBCs, they do not have the nucleus, rather that is why they are assuming a shape called as biconcave. So, they will be having a shape like this, so that they can easily move through the narrow capillaries. Okay. So, this is one interesting thing about the RBCs, and contains iron containing protein. I told you they are having hemoglobin. So, in the last respiratory system we learned, isn't it, RBC contains hemoglobin. This hemoglobin is helping in transport of blood from one part of our body to another, the gases, right. So, that is oxygen and carbon dioxide. They are biconcave in shape. I told you biconcave. So, this is biconcave. Okay, this side also, this side. So, biconcave in shaped, and a healthy individual has 12 to 16 grams of hemoglobin in every 100 ml. So, healthy person will be having this much amount of, how much? 12 to 16 gram of hemoglobin. And when you are moving on to the lifespan, if you see how long the RBCs can survive, they can survive to about 120 days. Okay, so the lifespan is 120 days. After they die, the RBCs are collected to special structures called as spleen. What is the structure called? It is called as spleen. In that spleen only what is happening? The RBCs are destroyed. Okay. So, there are structures called as the spleen, which is helping in destroying the RBC. Okay. Where is the spleen present? So, when you are saying where the spleen is present, so you know the rib cage, isn't it? So, imagine this is the rib cage and this one is the stomach. On the left abdomen, just near the stomach and near the rib cage junction, you can find this organ is present, which we call it as the spleen. And what is the function of the spleen? They are acting as a graveyard of RBCs. Means the RBCs are destroyed, means there they are broken down and it has been converted into some other forms and is eliminated out from our body. Okay. So, that is regarding your erythrocytes. You can see the structure. This is the structure of erythrocytes. They can easily move through the blood capillaries because of their concave structure. Okay. Next, we are moving on to leukocytes. Leukocytes, WBCs, they do not have hemoglobin, that is why white in color, is colorless. So, that is WBC. Nucleated means they contain nucleus, they are comparatively less than RBCs, like you can have only how much, 6,000 to 8,000 per cubic mm of blood only. What is present? This leukocytes are present. Now, these leukocytes are divided into two types, which we have already discussed. One is the granules and another one agranules, means one is having the granules, okay, and another one is agranules, they do not have the granule. Okay. So, if granule is present, we call it as granulocytes, and if granule is absent, we call it as agranulocytes. So, leukocytes are divided into agranulocytes and granulocytes. Agranulocytes, when you are seeing, they are of three types. What are they? Lymphocytes, monocytes, okay, and lymphocytes and monocytes only. Basophil, you have to bring it this side. Okay. So, granulocytes, when you are seeing, it is neutrophils, eosinophils and basophils. Okay. So, neutrophils, acidophils, eosinophil can also be called as acidophils, and basophils are granulocytes. Okay. So, basophil, sorry, I have written this side, you have to bring it over here. So, the major composition is also given, how much is present, you can find. Lymphocytes 20 to 25%, monocytes 2 to 3%, neutrophil 60 to 65%, eosinophil 2 to 3%, basophil 0.5 to 1%, and the structures for each and every leukocytes are also mentioned. So, granulocytes do not contain granules and granulocytes contain granules. Okay. Now, neutrophils and monophils, when you are seeing, they are phagocytic in nature. Phagocytic means what? Imagine this is my blood vessels and into my body some germ is coming. So, immediately from the blood capillaries you can find the WBC will jump out, and thereafter they will move towards the germ. They will engulf the germ. Engulf means what? WKC can easily move towards it like amoeba. They can move towards it. Okay. After moving towards the germ, what they do is that they engulf it. They will digest the germ inside and destroy that foreign organism which are entered into my body, thereby preventing me myself from different infections. Okay. So that is the function of neutrophils and monocytes. Basophils, when you are seeing, when these reactions means after engulfing, secreting enzyme, digesting the germs and all, when you are seeing for those things, some enzymes are responsible. Some reaction should take place. Wherever there is an injury, germs are entering, some sort of reaction takes place in my body. For that reaction, in order to neutralize the germ or in order to kill the germ, what is needed? Some substances are needed. So basophil is helping in secreting histamine and serotonin, heparin, like these materials, which helps in what? Inflammatory reactions. So there you can find the reaction which is going on in killing the germs. For that, some raw materials are needed, that is being secreted by whom? This basophils. Eosinophils, what they are doing? They are resisting infections. Resisting infections means like to particular substances. If you are allergic, what will happen is that immediately when the substance get into your body, the eosinophil starts secreting some chemicals which will help in what? Means neutralizing that allergic substances by giving you some symptoms, isn't it? So he will start hutch or running nose will start. So like the germs are moving or the thing is moving out of my body. Okay. So for that, what and all is needed is being carried out by whom? This eosinophil. And lymphocytes, main important. This lymphocytes can be two types. One is B lymphocytes and another one is T lymphocytes. B lymphocytes are produced by bone marrow, and thymus gland is producing what? The T lymphocytes. So there are thymus gland producing T lymphocytes and B lymphocytes are produced by whom? This bone marrow. And what is the function of this lymphocytes when you speak? You know, the antigen is the germ which is getting into my body. So what is getting into my body? Antigen. So imagine this is the antigen which is getting the germ getting into my body. My WBCs, especially these lymphocytes, they will produce antibodies. What they are producing? They are producing antibodies. Now this antibodies will fight with the antigen, and if this antigen is powerful, then who will win? This germ will win and they start to multiply. I will suffer from the disease. If imagine this antibody is powerful, then antibody will kill this germ, isn't it? So the antibody, when it kills the germ, the disease, I am not getting it. That's why those who are much immune, for them the lymphocytes production and function is powerful. So that's why whenever a germ gets in, they can deal with it. But for some people who are prone to illness, means their lymphocytes production as well as functioning is not that proper, because of which antibodies are not produced in much amount, because of which the antigen is winning and you are suffering from different types of diseases. Now listen, if this antigen is winning, then I have to get rid of the disease, right? So when I go to the doctor, they will prescribe me what? They will prescribe me antibiotics. Okay. So when you take this antibiotics, actually what is happening? This antibiotics will help in the production of antibodies. Now this antibodies will be produced and they will start killing the germs which has multiplied. Then after some time, within a week, you will be recovering from the disease. So you have understood what is the importance of antibiotics. Antibiotics helps in the production of antibodies. This antibodies helps in killing the antigen, that is the germ. Okay. So I will repeat again. Three terms we have learned. One is antigen, that is the germ. Antibody is a protein which is secreted by my WBC lymphocytes. And if I take antibiotic, then this antibiotic will help in the production of what? Antibodies, which will kill the antigen. Okay, so the antigen will be killed. So that is the role of what? This lymphocytes, right? So this is just, they have shown you the WBC, how does it look like, spherical structures, they are having nucleus, we have seen, isn't it? Platelets. So platelets are also called as what? Thrombocytes, and they are produced by specialized cells, tissue cells called as what? Megakaryocytes. Megakaryocytes, karyon means you know nucleus, megakaryocytes means nucleus which are bigger in size. Okay. So from those cells only what is produced? This platelets are produced. How much amount will be present? It will be 1.5 lakh, not million and all, it is only 1.5 lakh to 3.5 lakh per millimeter cube of blood or cubic mm of blood. Okay. And this platelets play a major role in what? The blood clotting by releasing many substances. Okay. So, that is the role of what? This platelets. So, you can say this is the structure of the platelets. To some extent everyone looks similar. Okay. Now, blood groups. Moving on to the next topic, that is blood groups. We will be learning how clotting of blood is taking place. Before that, blood groups. Blood groups, when you are seeing, I can categorize the blood groups into 4 main types. One is A group, another one is B group, another one is AB group, and the last one is O group. So, how many groups are there? A, B, AB and O. Okay. Now, there is a factor called as what? RH factor. What is factor? RH factor. This is a protein. RH is what? A protein which was first discovered in a monkey called as rhesus monkey. What is the name of the monkey? Rhesus monkey. That's why the name is given as RH factor. If this factor, if this protein, rhesus factor is nothing but a proteinaceous secretion. If this protein is present in one's body, then it is positive. If it is not present in the body, then it is negative. So, if a person is A plus, which means A, and he is having the rhesus factor. B plus means B and he is having the rhesus factor. B minus means what? He is not having that protein. That factor is absent. RH factor is absent. Understood? So, like that you have blood group A plus, A minus, B plus, B minus, AB plus, AB minus, O plus and O minus. A negative, B positive, B negative, A negative, A positive, O negative, O positive, AB positive and AB negative. So, these are the different types of what? The blood groups, right. Now, you should need to know how the things are dependent, who can give blood to, who can receive blood from. You have a common idea, I think so. So, ABO grouping. If you are looking into this ABO grouping, based on the presence or absence of two surface antigen on ABC, we name them as what? A or B blood groups. Plasma, when you are seeing, so you know in the RBC there is a protein and in the plasma again there is a protein. These two together play a major role in determining what type of blood you are having, blood group you are having. Okay. Now, the person who is having blood group A, listen, the person who is having blood group A, he is having a protein. That protein we are called it as antigen. Means a germ like structure, means a foreign substance which is present in the RBC, like that you consider. Means it is a protein, okay, which is present on RBC. In everyone it is not present. This A is present in someone, which means it is a factor which is present in someone and someone doesn't have it, isn't it? So, if the antigen is present on the RBC A, then the blood group is A. Then the blood group is A. So, if A antigen is present on RBC of the person, then his blood group is what? A. If A antigen is present on the RBC of the person, then the antibodies which is present in the plasma is anti-B. Anti-B means what? It is another protein which is anti-B in nature. I will tell you what is anti-B, okay, afterwards. Now, so where is this antibodies present? It is present in the plasma. Where is this antigen present? It is present in RBC, okay. So, RBC contains the antigen, okay, RBC contains the antigen and plasma contains the antibody. What is the antigen? A is the antigen. Here what is the antibody? Anti-B is the antibody. Now, this donor group, who they can do, means who can donate blood to, okay. A can donate blood to A as well as, when you are seeing, means donor, those who can donate blood to this A, okay. A can donate blood to A, O can also donate blood to A. I will tell you first, just listen what I am saying. Blood group B, which means what? Antigen which is present on RBC is B, that's why the blood group is B. So, if antigen B is present, then in the antibodies what will be present? Anti-A. So, he can get donor, who can be donor of this blood group, means a B blood group person can donate to him as well as a O blood group person can also donate him, the B group, okay. Now, third blood group is your AB. What is the third blood group? It is AB, okay. This AB, when you are seeing, they are having both the antigens on the RBC, that is A antigen is also present, B antigen is also noted. That's why the blood group is what? AB. Now, when you are seeing antibodies, if it is present anything in the plasma, no, they do not contain any antibodies. So, who and all can donate blood to this AB when you are seeing? AB can donate blood to AB, A can donate blood to AB, B can donate blood to AB, O can also donate blood to AB. Since this AB can receive blood from every one of them, isn't it? AB can receive blood from every one of them, whether it is A no matter, B no matter, AB no matter, O no matter, which means that everyone can give blood to this AB. That's why this AB blood group is called as what? The universal recipient, receiver, universal receiver, can receive blood from anyone, no matter, okay. But we have to consider another one factor, but blood groups when you are considering, A can give, B can give, AB can give, O can give, everyone can give, but positive and negative we have to see, okay, afterwards. Now, another thing is your O blood group. If a person is having O blood group, which means in his RBC there is no antigen, no, zero antigen, means nothing is present, no protein is present in his blood, RBC, okay, of blood. But antibodies, when you are saying, he is containing both the antibodies are present. Anti-A is also present, anti-B is also present. Now, who can give blood to O? Only O can give blood to O. Others cannot give. Now, listen, I will tell you why others cannot give. Listen, A is the blood group. I will just explain. A is the blood group, okay. Why B cannot give? Listen, if the B blood group person is giving, okay, B is a new type of antigen which is getting into the body. Now, when this B is getting into the body, A blood group person, what he has anti-B. Anti-B means what? Antibodies to kill B, isn't it? So, this antigen, if the B blood group person gives to A, what will happen? This B blood group contain antigen B. So, if this antigen B goes into the A blood group, anti-B is present where? In the A blood group. Yes or no? In the A blood group you can see what is the antibody which is present? Anti-B. Now, this anti-B will react with this B antigen. They will fight and the whole blood system will collapse and it will lead to death. Yes or no? That's why B cannot give blood to A. The same only A cannot give blood to B. Why A cannot give blood to B? Like A is giving, imagine B is the one which is present, B blood group, okay, he is having B antigen. A antigen is entering. So, in his plasma what is present? Anti-A. So, this anti-A and A will react, and anti-A, what will happen is that the reaction will lead to the death of the person. So, A cannot give blood to B, okay. Next you see AB. What is the advantage of this AB is that AB, when you are seeing, they are having both. Anti-A is, antigen A is also present, B is also present. If A is giving blood to this AB, so AB is the patient, okay. Imagine if A is giving blood, whether there is any problem? No, because A is already present in it. So, if A is giving, no problem, he is already having A, so it is not like a foreign substance. And if B is giving, there is any problem? No problem. B is also present in his body. So, he will not be having any problem because he is not having any antibodies to fight against A and B. So, that AB is what? A universal recipient. Another one thing is that I told you O can also give blood to AB. O can also give blood to AB. Why? So, AB also can give, isn't it? AB can give, no problem. O can also give. Why? Because in O, whether there is any foreign body present, any antigen is present, no. So, if O is giving blood, which means there is no A, no B. So, what is the matter? No foreign body is present, only the blood is being supplied without A and B. O is not having either A nor B, isn't it? So, automatically O is a universal donor. O can give it to anyone because in his body there is no A antigen or no B antigen, okay. But problem with O is that O is having what? Anti-A and anti-B, which means O can give to everyone, but O cannot receive from anyone, because if A is giving, he is having anti-A, if B is giving, he is having anti-B. If AB is giving, in that case also anti-A and anti-B is present. So, what will happen? It will lead to the death of the person, isn't it? So, O can give it to everyone, but O cannot take it from anyone other than O himself. O can give blood to O only. Okay. So, universal recipient is AB, means universal we can receive from anyone. That blood group is what? AB blood group, okay. But AB blood group can receive from any person, no problem. But O blood group is a universal donor. O can give, it's a rare group, and O can give it to anyone because it is a universal donor, as it does not contain any of the antigen A or antigen B. Is that clear? Other than this, there is another factor I told you, RH factor. This RH is an antigen, a protein which was discovered where? In this rhesus monkey, on the surface of that monkey it was observed, and those who are having this protein is RH positive, and those who are not having this protein is RH negative. Now listen carefully, RH negative person, if positive is getting into, will have what? Collapse, means they will lead to mismatch, compatibility will not be there of the blood, and the person can die. Okay. So, RH negative, when you see, listen very carefully, I will say once only. So, RH negative can get blood from RH negative. Okay. RH negative can give blood to what? RH negative, no problem. But RH negative cannot get blood from RH positive. Why RH positive? Because positive is having this antigen. So, this antigen is a foreign body, isn't it? So, if it is given to this RH negative people, so B positive is giving to B negative, not possible. Why? Because this B positive means which contains that person is containing a antigen. It is a different protein which is present in that person. So, that B negative person is not having that antigen. So, positive cannot give it to negative, but negative can give to positive. Why? Because listen carefully, negative can give to positive. So, RH negative person can give to RH positive. Why? This RH negative means they are not having any protein. So, no new substances is going, only the blood is being transmitted, isn't it? So, RH negative can give to RH positive, but RH positive cannot give to what? RH negative. This also you have to keep in your mind. So, when I am going for checking whether I can get blood from some, first I will check whether AB or O blood group. Thereafter I will decide what is my blood group type. Then I will see whether it is positive or negative. Thereafter I will go for donor checkup. Then if everything is matching only, the blood transfusion will be done, otherwise blood cannot be given. Okay. So, this is regarding your RH grouping. Now, the next point what we are going to discuss is a condition called as erythroblastosis fetalis. Listen ma, this is a condition which is noted because of this RH incompatibility. Okay. So, RH factor, based on that there is a condition present called as what? Erythroblastosis fetalis. Now, listen very carefully. Mother is RH negative. So, what is mother? Mother is RH negative, means she is not having any antigen. Okay. She has conceived her first child who is RH positive. So, first time she has conceived and she is carrying a baby who is RH positive, but the mother is what? RH negative. Okay. Now, what will happen is that since the placenta is present as a means a block between the mothers as well as the what is that baby, the fetus, you can find that they are not, the bloods are directly not coming in contact with one another. Okay. So, no problem, blood when it is not coming with contact with one another, it is fine. The first baby will be born safe, no problem at all, the first baby okay will be born safe. Now, listen carefully again. But while this first baby is born, at that time you can find cutting, umbilical cord, blood and all will be diffused. In that case, some of the blood from the baby while giving birth would have mixed with the blood of the mother. Okay. So, what? Well, while giving birth means at the time of giving birth, some of the blood of the baby would have mixed with the blood of the mother. because placenta everything is coming out, the blood is being evolved, umbilical cord is cut. So, you can find some mixing is taking place between the mother's and the baby's blood. So, some amount of this positive factor, what is moving into this positive factor is moving into the mother's blood. Yes or no? Since it is some amount of positive, which very less only it will be moving, the mother's blood will start producing, so antigen is moving, isn't it? So, positive means one antigen has moved into the mother's blood. Now, this mother, what they will do is that they will start producing antibodies. What they will start producing? Antibodies. So, one antigen, a foreign body is moving into mother's body while giving birth from the fetus to the mother, the positive factor. Now, this positive, Rh positive factor has moved. This positive factor, since it is very less in contact, the mother will not die. Okay. So, mother will instead start producing what? Antibodies against this positive factor. Now, the antibodies are present in the mother's blood. Okay. Now, if she is conceiving again, understood? So, if she is conceiving again, so what will happen? And again that baby is Rh positive. Imagine that baby is Rh positive. In that case, what will happen? Mother's blood is having the antibodies ready, isn't it? Against that Rh positive, antibodies are made by the mother now. It was previously not there, first delivery. In first case it was not there, but second time when you are saying the antibodies are present. So, immediately when the fetus is growing up and production of blood is starting, you can find the mother will start, this antibody production will be there, already it is there. So, antibodies will move to the fetus, okay, diffuse into slowly and they will kill the fetus. Okay. So, like this the fetus, the second delivery is not possible, the fetus may die. So, what usually doctors do is that, so Rh negative people, immediately after the first delivery, they used to give what? Anti-Rh, medicines. Okay. So, in that case, the mother will not start producing what? The antibodies, so that if the second fetus is positive also, there is no other issues. If it is not given, then what will happen? The second time when she is conceiving a positive child, then it may lead to the death of the child. The condition is called as what? Erythroblastosis fetalis. I will read out now. A special case of incompatibility between pregnant mother and fetus. Okay. When the mother is Rh negative, this is the condition. Okay. Rh negative, mother should be Rh negative and the fetus is what? Rh positive. The Rh antigen of the fetus do not get exposed to the Rh negative blood of the mother in the first pregnancy, as they are separated by placenta. But during the delivery, there is possibility of exposure of maternal blood to small amount of Rh positive of the fetus. Okay. So, maternal blood and Rh blood of the fetus, positive blood of the fetus are coming in contact. Mother is starting to prepare what? Antibodies against Rh blood group. Okay. Now, in case subsequent pregnancy, means the next time she is getting pregnant again, and the Rh antibodies from the mother can leak into the blood of the fetus. Okay. There is a chance that it can leak into, while giving food, while giving oxygen to the fetus, sometimes these antibodies can leak into. Okay. So, these antibodies are very minute, which can dissolve, easily pass, diffuse through the membrane and they can reach the fetus. If that is happening, then the fetus will suffer from severe anemia and jaundice and can lead to the death of the fetus. That condition is called as what? Erythroblastosis fetalis. Okay. This can be avoided by administering anti-Rh antibodies. I told you na, immediately after first delivery, what has to be given? Anti-Rh antibodies should be given to the mother, so that she is not producing antibodies against positive factor. Okay. So, this is regarding your Rh factor incompatibility, which is called as erythroblastosis fetalis. So, mother is negative, father is positive and the baby who has been conceived is positive, mother is negative, right. Now, in the first delivery, no problem, the baby has come out, but that positive factor from the body has moved into whom? It has moved into the mother. So, mother is starting to produce this green color is what? Antibodies. So, second time when she is again pregnant and she is having a positive child, then this antibodies will go and kill the antigen, that is the birth of the baby, means they will attack the red blood cells of the fetus and the fetus will die. So, this condition we call it as what? Erythroblastosis fetalis. Okay. The last part of the chapter is coagulation of blood. So, this is how the blood is clotting. Okay. So, how is this clot formed? No, one more thing is there, lymph is there. Okay. Anyhow, so clotting of blood, listen carefully. When there is a cut, an injury, what is there? There is a cut or an injury. So, in the injured side, imagine this is my hand part and here the cut has taken place. So, in this place you can find their tissues are getting injured, as well as there are some blood vessels. So, that blood vessel is also getting injured, isn't it? So, the platelets present in the blood vessels are coming in exposure to air. The injured tissues are also exposed to air now. Now, what will happen is that they will start, they will start releasing a substance called as thromboplastin. So, it is a proteinaceous secretion called as what? Thromboplastin. So, injured tissue, when it is injured, they will release what? Thromboplastin. Platelets, when it is when there is an injury, the platelets, when you are saying, they are releasing a factor. They disintegrate, means they dissolve and they disintegrate, break, and they will release a factor called as platelet factor 3 or platelet thromboplastin. These two thromboplastin, in the presence of, in the plasma of the blood, in that place the plasma is present, blood plasma, the fluid content of the blood contain calcium and some other proteins. So, in the presence of this calcium and proteins, this thromboplastin will release what? They will release what? Thrombokinase or prothrombinase. So, thromboplastin in the presence of calcium and protein ions are converted into what? Thrombokinase or prothrombokinase. Now, this prothrombokinase or the thrombokinase, it inactivates heparin. Heparin means what? Heparin is an anticoagulant. Anticoagulant means they will not allow the coagulation of the blood. Why? Inside the body, this heparin is there in the blood. If the heparin is not there, if I am pinching someone in that place, so imagine this is my hand, I have pinched in that place. So, inside maybe some tissues are damaged. If this heparin is not there, what will happen? In that inside, what will be formed? A clot will be formed. Dangerous or not, isn't it? So, there is a substance called as what? This heparin. This heparin, what will happen is that will get inactivated when what is released? Thrombokinase is released. So, this heparin gets inactivated, and in the presence of calcium ion in the plasma, there is another one protein present called as prothrombin, which is converted into thrombin. So, thromboplastin is releasing thrombokinase enzyme. This thrombokinase is converting prothrombin into thrombin. Thrombin is activating, again this is an enzyme which is converting fibrinogen into fibrin. Now, this fibrinogen, when you are seeing the fibrin, blood cells all combined and they are forming this fibrin, is a thread like structure where all the blood cells are coming and joining, and they are forming what? They are forming a clot. I will repeat again, listen carefully. So, where there is an injury, platelets and the injured tissue release thromboplastin. This thromboplastin, in the presence of calcium and proteins and calcium ions, are releasing what? Thrombokinase or prothrombinase. It is an enzyme. It is inactivating heparin and they are helping in the conversion of a prothrombin which is present in plasma through thrombin. This thrombin will help in the conversion of fibrinogen into fibrin. This fibrin is a fiber like structure. There all the blood cells will go and attach themselves and results in the formation of a clot. So, you can find this is a blood clot, this is the fibers which are present, fibrin fibers. So, for the formation of that fibrin, the platelets are playing a major role. So, like this only what is taking place, coagulation of blood. So, thromboplastin in the presence of calcium protein, thrombokinase is converting prothrombin into thrombin, thrombin is converting fibrinogen into fibrin, and the fibrin or the fibers, they collect all these cells, they come and get attacked, they attached there and they form a clot. So, this is regarding your blood clotting mechanism, a very important 3-mark question. You have to draw this schematic diagram, okay, representing coagulation of blood. The last part for today is your lymph, okay. So, lymph, when you are seeing, what is this lymph? I told you, lymph is a fluid which is present along, blood is one fluid, another fluid which is present is the lymph, okay. So, this lymph is present, this lymph fluid when you are seeing, where and all blood capillaries are there and all you can find another one system, blood vascular system is there is another one system called as what? The lymphatic system. This lymphatic system is present, distributed such a way that where blood and all cannot reach, even there also what can reach? This lymphatic vessels, lymphatic capillaries can reach. So, each and every places in our body, each and every tissue, cells where and all is present, from there the fluid is being extracted and it is taken to the lymph. Through the lymph the fluids are moving. Each and every substances from one part of our body is carried to some other parts either through the blood or through the lymph, okay. So, since it is present in between the tissues, we call them as what? The tissue fluid. What do we call them? Tissue fluid or interstitial fluid. So, at the side of capillaries, some water along with the water soluble substances move out into the space. So, this is a capillary, some water, other substances move out of the blood capillaries and they are collected between. So, cells and all are present between the cell spaces, there they are getting collected, okay. This fluid we call it as what? The lymph or the tissue fluid. Here only actually between the cells what is taking place? Exchange of gases, exchange of substances are taking place, okay. So, the lymphatic system which is present near to this what is that cells and all between the spaces, there is, I told you, there is a system present called as what? The lymphatic system. They collect all these waste materials, things and all, and they drain it to the what? They will take and bring it to, give it to the blood, okay, through the veins they will give. So, they are collecting. So, these lymphatic vessels what they are doing? They are collecting from the tissue fluid, they are collecting the materials which has to be excreted out and all. They are collecting and they are supplying it to what? They are supplying into the veins. Veins, you know, blood vessels, when you are seeing, there are veins, there are arteries and there are capillaries, isn't it? Capillaries are small. So, to the veins they are draining this out. Lymph, when you are seeing, it is colorless in nature and they contain a special type of lymphocytes and they play a major role. Since I told you where and all our what cannot reach? The blood cannot reach, the lymph can reach, which means they help in immune responses. Immediately when there is an attack by some germs, they play a major role in attacking along with the WBCs. Lymphocytes are present in the lymph and they do not have what? The lymph do not have RBCs. That is why they are colorless in flow, okay. Lymph also help in what? Absorption of fat. So, when you are seeing digestion, the digested particle, the amino acid of protein is taken by the blood, glucose is taken by the blood, but fatty acids cannot be taken by the blood. It is carried by whom? This lymphatic system, okay. With this I am stopping for today. Hope you have understood. If any doubts, please feel free to call me. Thank you.